Welcome students. Title of the unit is Unit 1 Introduction to Plant Tissue Culture. Module name is Plant Tissue Culture Laboratory Design and module number is 03. The outline of my module is the concept of the plant tissue culture laboratory and the laboratory design. The learning outcomes of my module will be, this module will explain the concept of plant tissue culture laboratory. It will describe the layout of the plant tissue culture laboratory, and it will also explain the importance of different rooms which are allotted in the laboratory. So what is plant tissue culture? Plant tissue culture refers to the in vitro cultivation of all plant parts, whether a single cell, a tissue, or an organ on a nutrient media under aseptic condition. What do you understand by this in vitro cultivation? Let me tell you, there are two types of cultivation or research which are done. One is in vitro and one is in vivo. In vitro cultivation is nothing but the research which is carried out outside the body of a microorganism or a plant. And in vitro and in vivo cultivation is a work or the research which is carried out inside the body of a plant or a microorganism. So what is this nutrient media? Nutrient medium is nothing but growth medium. It can be either be solid or it can be liquid. This medium supplies with the nutrients which are required for the plant to grow. And what is this aseptic condition? Aseptic condition is a condition which is free of all kind of germs. These germs include microorganisms like bacteria, fungi, and viruses. So why do we have this plant tissue culture which is in vitro? This is done to have plants which are disease eliminated, which are virus eliminated, for the production of secondary metabolites. As you all know, some secondary metabolites are used in pharmaceutical for the production of different medicines, and this is also used for crop improvement. So basically in vitro cultivation of plant cell is used to keep the plant tissue culture contamination free. So let's go ahead with the design of plant tissue culture laboratory. The tissue culture laboratory includes the following rooms. There are 4 rooms which are involved. First is the washing room, second is the media preparation room, third is the transfer room, and fourth is the culture room. Let me tell you all, a plant tissue culture laboratory is a closed system type laboratory in which the external environment doesn't figure with the internal environment. This is done to avoid the source of any type of contamination which will cause harm to the plant tissue cultures. Let's go in detail with each room. First room is the washing room, in which we have the wash basin, we have the tap water system, we have the water distillation unit, and we have the hot air oven. The wash basin which is kept in the washing room should be made of stainless steel so that it doesn't react with any kind of acid and base. Next we have the tap water system, so that we have both the outlets, the cold water outlet and the hot water outlet. Next we have the water distillation unit, which supplies us with distilled water, which is further used for preparation of media and washing of glasswares. And fourth we have the hot air oven, in which we keep all our washed glasswares for drying. Before we keep the glasswares for drying, we ensure that the glasswares are washed properly. For washing of the glasswares are soaked in a detergent overnight, and then they are washed thoroughly with tap water, and then they are rinsed with distilled water, and then they are kept in hot air oven for 150 to 200 degrees Celsius for 1 hour. Care in this room should be taken that solid media which is used for culturing should be discarded immediately. The contaminated media should be decontaminated first using an autoclave and then kept for washing. Now let's go to the next room, which is the media preparation room. In this room you will find the working benches, chemical reagent, glasswares or plasticwares, and the instrument. This room will have ample amount of working space, and it will have ample amount of storage space. This storage space is used to store chemical reagents, to store glassware and plasticware, and also to store double distilled water or distilled water which is made in the previous room. This room will have various kind of instruments like the weighing balance, the pH meter, the refrigerator, the microscope, the autoclave, etc. In this room, the care should be taken. You should have time to time or day-to-day record of the usage of the instrument. The chemical should be stored alphabetically. The highly volatile or strong acid or base should be kept separately, and chemicals which require low temperature should be kept in the refrigerator. The next room is the transfer room. Initially, we used to have a small room which is fitted with a UV light above and bacteria-proof high-efficiency particulate filters. Now, this is reduced and it is made into a small instrument which is known as lamina airflow cabinet. This lamina airflow cabinet has HEPA filters attached to it. These HEPA filters can be two types. This lamina airflow cabinets can be two types. One is a horizontal type and one is a vertical type. In the horizontal type, the HEPA filters are placed horizontally, whereas in the vertical type, the HEPA filters are placed vertically. This unit will also have a provision of UV light, a provision of normal tube light. It will have a working space which is made of stainless steel. Prior to the work to be taken, you have to sterilize this instrument. The stainless steel working bench is sterilized with 70% alcohol, and after that the UV light is on for 15 to 20 minutes. After this time period is done, you have to switch off the UV light, you have to switch on the normal light and the air blower, and then start your transfer experiment. This room will have double door system. We should ensure that there is no opening in this room which is kept to avoid any sort of contamination. Now the last room which is present is the culture room. In culture room, we have kept the culture bottles and culture tubes under controlled condition for incubation, like the temperature, light and humidity is controlled. The temperature is kept at 25 plus or minus 2 degrees Celsius and the humidity is controlled above 50%. In this room, there are different racks which are made. They are of stainless steel. Each rack has a tube light which is attached. This tube light is attached with an auto timer to give photo period to the culture bottles. This room has highly controlled temperature and humidity. It has a thermometer and a hygrometer which is kept in the corner of the room. One should see that they should avoid wearing their footwear inside this room to avoid any kind of contamination. So let's summarize this module. Plant tissue culture is a collection of experimental methods of growing a large number of isolated cells or tissues under sterile and controlled conditions. To carry out the experiment using tissue culture techniques, a well-equipped laboratory is required. An ideal tissue culture laboratory should have a separate room for general laboratory and for aseptic work. These are some references which I have referred. Thank you.